I'm really pleased to be with you at our sunrise session this morning. I believe that there in the audience today there's probably some experts in their own right on the subject, and also others who are leaders in the community, business leaders and the like. And so what I hope to have for you today is some information for all of you. The subject is emerging worldwide infectious diseases, opportunities and prospects. How could that be? How could infectious diseases be opportunities and prospects? So I would like to explore that. Now, in 2001, September 11th, the infamous date, the federal government realized, and experts, medical experts realized, that the threatening agents, bioterrorism agents, many of these things, we did not have vaccines, therapies, or suitable diagnostics. And so the federal government charged the National Institutes of Health, and more specifically, National Institutes of Allergy and Infectious Diseases, to develop these therapies and vaccines and diagnostics. And so the people at these institutes decided an approach would be to gather together probably a group this size, experts in the field, scientists who would provide input to the federal government and develop a strategy so that they could move forward with this important task. And this is a document that emerged from that. And interestingly enough, with this strategy was not only to address infectious agents or toxins that are considered bio threats, terrorism threats, but also emerging diseases such as influenza or SARS. And so this group of scientists, now is this adequate enough to hear? Okay. This group of scientists categorized the various agents into categories A, B, or C, with A being the ones that had the greatest potential of threat, and C with a lesser potential of threat. And you can see some of these viruses. I've only listed the viruses because that's all the work we do. We do not do bacteria or toxins. You can see smallpox or dengue, Ebola, West Nile, rabies, SARS, influenza. And so you can kind of get an idea of the type of infectious agents that are in these groups. Now, I have a quiz for you this morning. And considering what happened on September 11th, but also other events that have happened since then in the world of infectious diseases, the quiz question would be, which viral disease has the greatest probability with the greatest threat or harm? Okay, smallpox, Ebola, hepatitis, West Nile, yellow fever, influenza. Don't raise your hands. Just write it on your napkins and I'll grade them afterwards. Put your name on the napkin. Well, I'm going to submit to you that the one that has the greatest probability is influenza. When smallpox was on the earth and spread through humanity as a disease, there were 3 to 5 million people a year that died from smallpox. Yellow fever, for example, has 30,000 people that die a year. West Nile virus in the United States, less than 150 people. Dengue, Lassa fever, Argentine hemorrhagic fever, all of these things are on that list. You'll notice that seasonal flu, the influenza that we have every year, approximately 500,000 people worldwide die from this disease. And you can see that there have been a number of epidemics through the years, or pandemics, if you will. Russian flu, Asian flu, Hong Kong flu, and about 1 million people had died each year from these outbreaks. The infamous Spanish flu, which I'm sure you probably heard of, in 1918, 50 to 100 million people died. Some people consider this the greatest medical holocaust in history, as illustrated by these couple of facts. This Spanish flu killed more than the total number of soldiers killed in World War I, happening at the same time. And this number was equivalent to the third of the population of Europe. And many people, historians and others, feel that this epidemic contributed to the Treaty of Versailles, and probably because the soldiers, many of them, were too sick to engage in warfare. Now, I found this photo in Utah State University archives. It's true. This is the 1918 Utah Agriculture College basketball state champions. And I show this for two reasons. One is to emphasize that we do have good basketball teams at USU. And the second reason is because I noticed that there are only 5 players on this team, and it's in 1918. I just wonder perhaps the others were sick with influenza. But the third thing, I suppose, is that maybe that's the reason why they won the state championship. The other teams were sick. Oh, no, that's not true. Somebody, you disagreed with that. Okay, they're just better. Okay, good. Well, what's happened since 2001? We now have become familiar with avian flu, more accurately referred to as highly pathogenic avian influenza. The 1918 flu, 1 out of 50 people would be expected to die. The HPAI, or avian influenza, if a person gets infected, a 50% mortality rate. It's extraordinarily high. And so if this were spread through the human population, it would kill hundreds of millions of people. Can it happen is the great question. And I can't answer that, but let's just look at 2 things. There are 2 key factors, in my estimation, that control this question. The first is the pathogenicity. How lethal is it? Well, we know that if a person gets infected with avian influenza, the highly pathogenic strain, that it is highly pathogenic. The question is, what about its transmissibility among humans? It is not very transmissible. And so this is what limits this potential biological holocaust. Is it possible then that this virus could become more transmissible and therefore retain its pathogenicity and cause a terrible disease worldwide? You might find it interesting that the receptors for this virus are somewhat, the cellular receptors for this particular strain of virus are somewhat different than the seasonal flu influenza. And in that this virus uses receptors that are deep within the lung and not higher up in the lung. And as a result, when it does infect the lung, it is very, among other reasons, it is pathogenic. But also it means that it's more difficult to transmit the infectious agent from person to person. Well, the question that I've had and others have had is, if this virus mutates to infect higher portions of the lung, its transmissibility may increase, but its pathogenicity may decrease. We just don't know then, in the end, what would happen, but we have to plan for it. At Utah State University, we have the Institute for Antiviral Research. These are the principal investigators at the Institute, and I would like for one of these, Justin Julander, to stand, please, if you don't mind. Justin was kind enough to come down and to support us here. We appreciate it, Justin. These investigators have different, skills, experience. Besides working with these various types of viruses, they also have experience with biosafety labs, biosafety containment, immunology, virology, neurovirology, and chemotherapy. And so the rest of the presentation, I think you can keep in mind the message that I would like to convey, and that is that if you gather together a group of people with different experiences and different expertises, and you have them work on similar problems, then you have a synergistic combination that results in important, I think, discoveries, and that's what I hope to show here. So what is the Institute for Antiviral Research? It was established in 1977. Since 1985, the Institute has brought in more than $46 million in federal funding. The majority of that funding is from the National Institutes of Health, and there's been some funding from early funding, I would say, with viruses of military concern from the Department of Defense, concerns about soldiers being exposed to exotic viruses and what do you do about that, and also some private industry. So you can see here are some of the viruses and studies that have been funded through the years. I will be talking about influenza, a little bit about mad cow disease, only because that's, I think, of interest and, to some of us, and then West Nile virus. Now, I'd like to tell a story here with a drug called T705. T705 was discovered to be effective in cell culture against influenza virus. Well, there's already a drug for influenza, and I'll talk about that in just a minute, so why would you need another one? Well, it turns out that this is very effective in both cell culture and in animal studies, and now it's in clinical trials. Don Smee has provided much of the data that has been used by Toyama Chemical Company in Japan to have clinical trials in Japan and the United States. Much of the animal data and cell culture data has been provided by his group. Also, Brandon Terrell. Brandon, you're back here. He tries to be incognito. Stand up. Brandon's a Ph.D. student, and he has been working also on T705 and West Nile virus and other types of projects. Well, I show this not to panic anybody, but I want to emphasize that this drug is a very simple compound. It's a low molecular weight, has good bioavailability in people, hopefully, and also in animals. And when it gets into the cell, it metabolizes to all these different types of chemicals. Well, what's significant about that? The significant part about that is that this chemical right here resembles the basic building block of RNA. And it turns out that this chemical is an inhibitor of the virus genome synthesis, which is an RNA molecule. So it inhibits RNA polymerase, or the enzyme that makes the viral gene. Now, I found on the internet a really nice illustration. He's a scientist in Great Britain who consented to allow me to use this animation today. So when you see it, you'll see it's not the virus. It's not simulated for the virus, but basically what it does is that it simulates. I'll just say this for the scientists in the group. It simulates RNA transcription from DNA, but the animation will illustrate how the virus synthesizes its own genome, and then I'll explain how the drug inhibits this process. So that red thing right there would be considered to be the parent virus. This blue thing is the polymerase, or the enzyme that is taking in these little yellow building blocks, RNA building blocks, and then synthesizing this yellow strand. See those building blocks coming in, synthesizing the yellow strand of RNA. And if we use our imagination a little bit, that could be influenza. And when one of these little building blocks is T705 molecule, it's like putting a stick in the spokes of a bicycle. It just stops it in its tracks. Well, at USU, we have, in the past, been involved in the development of Tamiflu. You're aware that this is probably the main drug that is applied for influenza. And you may have seen ads that this is a safety information ad. But basically what they're saying here is, yeah, we do have Tamiflu, but the problem is when you start to feel sick, the clock starts, and you only have 48 hours before you can take that medication and have it work. So it's a timed thing. You've got to get into your doctor, and you've got to get that medication quickly. And if you let it go beyond that time, it's no longer effective. Now, if you get it and there's a household and maybe one of the children get it, you can give it as a prophylaxis to the other members of the family to prevent them from getting it, but the unfortunate person who has influenza and waited too long will still get the disease. So the remarkable thing about T705 is that you can delay treatment considerably longer than the Tamiflu. This is just shows the treatment of flu or influenza in mice with T705. Now this is percent effectiveness. So 100% effectiveness would mean that it's perfect in its protection, whereas 0% mean that it had no effect at all. This black line is the placebo, and over time you can see that its effectiveness eventually goes down to 0. Whereas if you administer T705 at 3 days, it has very good protection or efficacy, clear out. And if you administer it at 3-day delay, 3.5 delay, 4 days delay, it still has protection. And at 5 days, you start to see a diminishing of the protection. The point with this slide is that one of the strengths of T705 that is now in clinical trials, which we've generated, Dawn, SME has generated most of the data that has been submitted for FDA approval for clinical trials. And the hope is that it will do the same thing in people, and we can extend that window of opportunity for treatment for influenza. The other thing that's nice about it is that it's active against resistant strains, strains of virus that are resistant to Tamiflu, and that's a real benefit. Well, we realize from work with the people who chemically synthesized and did the initial cell culture experiments in Japan that this drug is an inhibitor of this RNA machinery that synthesizes the virus gene, and could it be that it is also active against other RNA viruses? Now, in the world of antiviral therapies, this would be a remarkable thing. Most most viruses do not have that benefit. So most drugs do not have that benefit of broad reactivity. And so Brian Gallen, who's at our group, had been working on some other RNA viruses, other types of RNA viruses, Lassa fever type virus, Hanta, Rift Valley fever type virus. These are bioterrorism interest viruses. And this drug, when administered 5 days after the initial challenge of the virus, was 100% protective, this T705. And likewise, at 36 hours or 48 hours, it is also protective against these viruses. So we were thrilled to see that it was cross-reactive to other types of viral, probably their polymerases also. What about other viruses that we work on? Justin Julander, who I just introduced to you, has done work with Western equine encephalitis. Some people say, why is that virus? It's so rare. Why would it be on this bioterrorism list? Because these encephalitis viruses can be, unfortunately, aerosolized. And when they're aerosolized, they get into the nasal cavity, infect the olfactory bulb, follow the nerve tracks back to the brain, and the person has fatal, lethal encephalitis. And so that's not the normal route. The normal route of infection is by mosquitoes, like West Nile virus. And so that's the natural route. And it limits its spread. But it's on the list because of potential aerosolization. Well, it's effective. Now, this drug treatment is effective. The problem is, really, as the disclaimer, I guess, is that you have to add considerably more drug for it to be effective. And so, really, the verdict is out as to how useful this will be ultimately. But nevertheless, the fact that it's broadly reactive against these other viruses is of great interest to us and others. And then Brandon, who you met back here, he's done work with West Nile virus and likewise has shown this to be active against West Nile virus. The problem is that once the virus gets in the brain, we have not found it to be active, and that's probably the time at which people go see their physicians. And so really, in the end, we don't know how useful this will be for some of these other viruses. Well, what's the implications of this drug? Now, why did I show this example? I showed this example because the discovery of some drug or some virus mechanism specific to some specific virus, we have found, this is an example, that one discovery led to another discovery, led to another discovery, and it has broader utility than we initially would have thought. So, as I mentioned, T705 is in clinical trials if it is effective and if it is safe and if it is approved by the FDA, then the possibility would exist that this drug could be used off-label usage, which is legal by physicians, for other indications such as some of these other viruses. I'd like to shift gears for just a minute. This is the virus that I work on, so I'd like to show you some of our research. This is West Nile virus. West Nile virus, as you may know, is easily transmitted from the mosquito to the bird, back to the mosquito, back to the bird, in this cycle of replication. Now, occasionally the mosquito will bite a human being or a horse, and it will cause the disease. The reason why mosquitoes can't bite these individuals and then transmit it to another individual, that doesn't happen, is because the virus load or titer or numbers of virus in the serum of these organisms here is not high enough. So the mosquito does not get the virus once it has been transmitted. But here the bird viremia or virus titer in the serum is high, and so there's a cycle of infection. Well, that doesn't matter if you happen to be the person who gets infected, because, in fact, I just from a show of hands, how many in here know of anybody who has been infected or had West Nile virus infection? Oh, gee, I would say probably about a fourth or a fifth of you probably. So the thing with West Nile virus is that people talk about its mortality, but about it may cause death, but that's not the greatest problem. I mean, if it's your family member who dies as a result of it, it doesn't matter what the statistics are. But the vast majority of people who get West Nile virus infection get neurological disease. West Nile virus infects the brain and the spinal cord to cause neurological disease. So this is an illustration of a man who had been infected by a mosquito, and he could hardly hold his arm up because the nerve innervation of the muscles was damaged enough that he couldn't hold his arm up very well. And they're doing a neurological test on this man. Well, neurological disease comes in basically two stages. The first stage is the acute stage, when the virus it has a fulminating replication in the central nervous system, and there's a lot of things happening. And then it eventually goes away, and then there's a chronic, lingering neurological disease. And those of you who raised your hands may be aware that a person who's infected can get over the acute infection, but boy, they just have this lingering problem. Some people very serious and some people not so serious, but it's a lingering problem. And that's what we've been investigating. And in order to do that, we wanted to use, we looked for neurological assays that are used by neurologists in human medicine. We decided to look at electrophysiology. And this had not been done before in animals to study this virus. But basically, let me show you how it works. Now, you got to pay attention to this one, okay? Those of you who are asleep in the back. This is a cross-section of a spinal cord illustration. And there are neurons in the spinal cord that send out axons through the limbs and other parts of the body to tell our lung to breathe, to tell our heart to beat, or to tell our legs and arms to move. And so the electrophysiological assay that we do is that we will stimulate the nerves in the leg. And then downstream from that, toward the foot, we will record the conductivity, electrical conductivity, that occurs in this nerve. Now, this is a cross-section of a bundle of nerves. These are cross-sections of these little black lines or axons. And so, when you stimulate here, at a low threshold, low voltage, you may not activate any of these axons to conduct, and you'll just get basically a flat line on your oscilloscope. But when you reach the threshold, as indicated by yellow, you start to get a little blip. And Then you increase that a little bit more of these axons are activated, but it's hard to detect. But then eventually you start getting more and more of these axons activated as you increase the voltage more and more, as you can see by the color code. And you can see this is an actual tracing of a normal animal. And you can see how eventually you reach a maximum activation where almost all of these things are activated. That can be put into a formula, and we call that the motor unit number estimation, or MUNI. That's the way the assay works. The reason why I spent such detail on that is to emphasize our finding, which is that 100% of the animals that were infected subcutaneously, which is probably approximating the mosquito bite, 100% of those animals had decreased MUNI, even though they actually looked relatively normal to the naked eye. And whereas you can see the uninfected animals had normal MUNI and the infected ones with the red line didn't. Remarkable finding, considering that not only in the acute phase of the infection they had suppressed MUNI, but even as late as 100 days. Well, we investigated this a little bit further to find that this may kind of change the way we think about West Nile virus. It may change a little bit of the paradigm with this infectious disease. What I mean by that is it's originally thought that the virus basically grows here in the acute phase and then dies, and then the resulting damage is the cause of the neurological sequelae or lingering problems. Well, we find that in these animals, clear out to 100 days, we find here's some tissues that are stained for West Nile virus by red. You can see there's axon bundles that are heavily stained with the virus, and then that virus presence is associated with the pathology observed in these tissues. So even though there's not a lot of virus, there's some virus, and it's causing lingering problems, and that really has not been described before. That has real implications for treatment. We have, in collaboration with Washington University in St. Louis, been investigating a special West Nile virus-specific humanized monoclonal antibody for treatment of West Nile virus. I won't spend a lot of time on this except to say that it has really some unusual characteristics. Unusual enough in that it gets into the brain at sufficient quantities. It can stop the virus disease if given early enough, even after the virus has entered the brain or central nervous system. It's a remarkable characteristic of an antibody, but yet it works. Here, at day 4 or 5, the virus gets into the brain. If you treat it this period of time, again, a similar type of chart where this is 100% effective, this is 0%. You can see the placebo is not effective, but the antibody is. Well, this is the data that has been generated. A company called Macrogenics is not bought from us, but bought from Washington University, St. Louis, the rights to this. And the data that we've generated has been used to get approval to do clinical trials, investigational new drug trials with this antibody. And that's ongoing at this time. Boy, we hope it works, but there are going to be some limitations to it. Well, the last thing I'll mention is mad cow disease. If you don't recognize it, this is a mad cow. Mad cow disease is referred to as a prion disease or transmissible spongiform encephalopathy. The people do not get mad cow disease. They can contract a disease that is the human form called Creutzfeldt-Jakob disease, or CJD. 1 in a million people will get this spontaneously. It's a very unfortunate disease. I won't ask in here somebody who has known people who may have had this, but I see some heads that are shaking yes. We had a secretary in our biology department whose daughter had it. Unfortunate disease. So anyway, it's unusual. It's not a typical infectious agent, but and it's a tough nut to crack in terms of therapy. No one's been able to do it. No one's been able to come close. We have some preliminary data I'm calling drug A, unfortunately. I can't talk about that a lot. Brandon is the one who suggested this. Okay? I said, Brandon, it's not going to work. I said, and he says, but we got some, you know, and I said, okay, well, if it's not going to work, but if you really want to do it. And it, again, percent efficacy, it looks like there's a slight ability for this drug, when administered after the infectious agent is well established in the body, either at 14 days administered or 28 days and then administered daily thereafter, to be efficacious. We'll see how that works out. I just wanted to show you that just to illustrate that we are working on this disease. This is a summary. Existing and emerging viruses continue to pose a significant threat. Utah State University, I hope that we've talked about some of these new discoveries, that you're convinced that we are doing some things in the field that have contributed, and we hope to contribute further. And so did you notice the mask? Our helper, she did that little, so hopefully we won't have to wear those masks, I guess. Okay, I'll go ahead and take questions, please. Thank you. Oh, just a minute. Excuse me. You have a question, but you're sitting next to somebody I didn't introduce. Andy's picture was up there. I want you to stand, Andy. Andy's a pre-med student. He's working in the lab for some time. He's been so good. Stand. I want to recognize. Stand. He's been so good. He's been so good in the lab that we've actually hired him full-time. He's still a student going through school. He'll finish up here pretty quick, hopes to go to medical school. Andy has contributed a lot to a lot of the testing for West Nile virus and cell culture and animal work. Really good, if any of you want to know. And if there's anybody here from the University of Utah Medical School Admissions Board, you can take note of that. Okay, now we have a question right here. That's a great question. One of the paradigms, I would say, that if you discover an inhibitor of RNA polymerase, which is what you're talking about, for a virus, it's going to be cross-reactive with cellular RNA polymerases. That's just the way it's worked out. And that's the reason why there have been no good discoveries on inhibitors of our viral RNA polymerases. Remarkably, this drug has very low toxicity for eukaryotic cells, and it has a very high specificity, broad viral high specificity for RNA polymerases, viral RNA polymerases, a remarkable characteristic. I suspect that we only know the tip of the iceberg as to the mechanism of this. I suspect there are other mechanisms associated with the observed inhibition of RNA polymerase with this drug. Yeah, Ned. Yeah, the earlier you would give T705 presumably, and even prophylactically, it would even be that much better than if you gave it at 3, 4, or 5. Yes, please.
Which brings up one question for me, and that is, and I realize you're working with animal models, so this might be a difficult question, but what would be the side effects?
The side effects of T705? Mm-hmm. I am aware of one particular side effect. I don't know that that's been disclosed or open from clinical trials, but it will not prevent this drug from moving forward if it's just that one toxicity side effect, but there is some toxicity associated with it. But the phase 1 clinical trials are in progress. The blinded studies have not been unmasked, so we don't know the real end product of that, but you know, maybe it'll be stopped in its track by some toxicity that we've not previously observed, and it will occur in humans. It will no longer be suitable for advancement as a real approved drug. We just don't know at this point.
If somebody takes a dose of T705, how long will it have a positive effect on the body? How long does it last?
The half-life. Yeah. That's what essentially you're asking. How long does it stay there? You've got to take it once a day. As well as Tamiflu and some of the other medications. Yeah. First off, The T705 is active against Tamiflu-resistant viruses. It's much more difficult to develop resistant strains of influenza to T705. In fact, it's really difficult, and there's some question as to how, so it's plus minus. Yes, sir. Yes, yes, poliovirus in particular, and it has activity in cell culture. It's not been tried in anything else other than cell culture. But I will say, gosh, we've got some good questions here, so I know that but poliovirus is a prototypic RNA polymerase virus, and it does have activity against that. It's not quite as good of an activity of a effective 50% effective concentration as with influenza, but it's pretty doggone good compared to some of these other viruses like West Nile virus or Western equine encephalitis virus. Oh, okay, yeah. Well, no, no, not really, because it's so expensive that you wouldn't be able to use that in cattle. Realistically, it would be prohibitive. Can you get any information from that that would help the livestock industry? I can't foresee something at this point, but who knows? Kent, you're about ready to put a stop to this fund, so.
Not at all.